We now have Professor Jack Heinemann. Jack Heinemann is a professor at the University of Canterbury, another very fine institution, specialising in the field of molecular genetics. He has won awards for his teaching and his research and published many research papers, has held academic posts in Norway and the United States, and served at the United Nations. Among Professor Heinemann's main interests are in biochemical and genetic characterisation of horizontal gene transfer, in particular how it relates to the evolution of antibiotic resistance in microbes. Please.
I want to say to the organizers, thank you for having me. It's a real pleasure to come to my own backyard and see Lincoln. Just before I begin, I want to do a little survey. How many of you have heard of COVID-19? Okay. No surprises there. COVID-19 is all but a pandemic, and for good reason. It has a high mortality rate, something on the order now of 3.4%. And we know this because its appearance has caused a global surveillance system to emerge. It emerged as recently as December 2019 with 106,000 deaths so far, which calculates as one death every 1.2 minutes. So it's an important and fast-traveling pathogen. But we have another pandemic, one that's been with us for decades. But we don't call it a pandemic. And that pandemic is the rise of antibiotic-resistant bacteria. Now, not as early and virulent as COVID-19, but we don't have a global surveillance system for antibiotic resistance. So we're only guessing at the numbers of people who are dying from antibiotic resistance. But even in really wealthy countries like the US, there's one death every 12 minutes. India, just with babies, 58,000 in a year, one every 9 minutes. If we had a global surveillance system measuring the deaths from antibiotic resistance, it would likely dwarf what we'll see from COVID-19. But we've become sort of complacent about the antibiotic resistance problem. Unfortunately, the pace at which we'll be able to control antibiotic resistance has also slowed. We don't have, on the horizons, new drugs to solve this problem. And even if we did, we have the ethical dilemma that we still haven't learned why the old drugs are failing. Is it because the biology is ultimately always going to overwhelm our drugs? Or is it because of the economic models through which we have now chosen to develop and then sell new drugs that leads to their obsolescence? So it becomes an ethical dilemma. Even if we were to find a wonder drug tomorrow, should we release it, knowing that we would probably doom it to failure and deny it for future generations? Another little audience participation. I'm going to give you the option, and you have to vote, and we're watching, okay, so we'll know. You have the option of swimming in one of two rivers. The first river has lots of E. coli in it, and there's a good chance that after swimming in it, you will have a gastrointestinal or skin infection as a result. Pretty good chance that itchiness is from the river. Here's your other choice. A river with many factors lower of E. coli and other kinds of pathogenic bacteria. So the chances of getting an infection are also much lower. But all of the bacteria in this river are resistant to antibiotics. So if you get one, there's a really good chance that itch won't be cured. Which river would you choose? Those of you who want the river with lots of microbes and a chance of really getting itchy, and those of you who would prefer to die of an antibiotic-resistant infection, but the chances are low of getting one. I am pleased to say I can cater for all of your desires. Right here in Canterbury. No choices. You have to swim in this scenario. You could drink it. I mean, if that's what you're offering. So drinking or swimming. So here's one of our studies. What you see here at the top is a profile of the Avon River running through Hagley Park in Christchurch. And what you see is, first and foremost, that we have pretty high E. coli counts in the Avon River, well above what we would normally call the safe swimming limit. And what you see also is that a lot of those E. coli are resistant to a lot of the antibiotics we measure. And these are not just random antibiotics, these are antibiotics that are still clinically useful for the control of E. coli infections, including the frontline antibiotic ciprofloxacin. On the bottom, you see a rural stream, silver stream. It has a lot of things in common with the Avon. It has high E. coli levels. And it also has E. coli that are resistant to some of the same antibiotics. But the frequency at which we detect those resistances is much lower. And the frequency at which we detect really high levels of resistance is much lower. So here's your choice. For the minority, you can go swimming in the Avon River, or if you're in the majority, Silver Stream, which is dominated by agricultural practices. This is just to say, though, that what this profile doesn't capture, the way in which we look at microbial counts in water, doesn't capture the risk factor of antibiotic resistance. Two rivers, each of which look equally dangerous from the point of view of the microbiological assessment, are fundamentally different from what the vast majority of you found important, whether or not those bacteria were resistant to antibiotics. And that is not what we monitor. We don't routinely monitor resistance. When we looked at these bacteria, we also found that they didn't tend to just be a little bit antibiotic resistant. They tended to be really antibiotic resistant. So what you're seeing here is a panel of, 8 different antibiotics, and the frequency at which we got resistance to those antibiotics. The darker the square, the more antibiotic was required to kill those organisms. That's the MIC, or minimal inhibitory concentration. You don't see very many light squares. And that's not because of the lighting in the room or how far back you are from the screen. It's because they're not there. You either get full susceptibility or you get untreatable. So these concentrations that they're resistant to are, in the main, untreatable in a clinic. We cannot give you enough antibiotic to clear infections from these bacteria. Here's a case in point of a couple of the strains that were taken from the Avon and the Silver Stream rivers. Many of the red arrow here at the left is the clinical breakpoint concentration. This is the concentration of ciprofloxacin your doctor can give you. Above that concentration, the antibiotic starts to kill you. So there is a real limit to how much antibiotic we can use. We routinely found bacteria that were resistant to concentrations of ciprofloxacin 32 times higher than can be administered to a patient. So why? Well, one popular and also true explanation is that we have resistance because we use antibiotics. And medicine is a big user of antibiotics. We have also underestimated the concentration at which antibiotics start to select for antibiotic-resistant bacteria. As little as 10 parts per billion of the antibiotic already begins to select differentially in populations of bacteria for those that are more resistant to antibiotics. But agriculture is the world's biggest user of antibiotics. So more than half of the world's antibiotics are distributed into the world via agriculture, so and directly then into the environment rather than into human beings. Up to 80% of an administered antibiotic in an animal comes out in the urine and the feces. And those concentrations are still above the minimum inhibitory concentrations, so that means they're already selective in the poo patties on the paddock. These antibiotics are then redistributed in our world in various ways, such as when we use them for manure. So it's not just that animals are exposed to the antibiotics. Our crops are also exposed to the antibiotics. And our crops are full of microorganisms. And our microorganisms on those crops have been selected for being antibiotic resistant. So far, that's not a big deal, because most bacteria don't cause disease in people, livestock, or pets. However, this increases the probability that exactly those kinds of organisms will also be exposed. We live in an increasingly chemically intensive world, with antibiotics being just one of the chemicals that we spread now nearly everywhere. In addition, are chemicals such as pesticides, and in particular, herbicides. So just here on the left is a table from a few years ago looking at the amount of various herbicides, the big group of herbicides used worldwide. Over 635 million kilograms of glyphosate-based herbicides are used worldwide. Each year. And in the table below is just stats from the United States alone with their big herbicides, glyphosate, 2,4-D, atrazine, and dicamba. Herbicides are used not just in agricultural environments, but also urban environments. So if you're going to be exposed to one of our most commonly manufactured chemicals, it's likely you're being exposed to herbicides. And it's becoming so common now that you find herbicide residues in waterways and also in people. It's routinely being detected in urine. So we know the exposures are high. There are many different ways in which we become exposed. For example, our pets can be on antibiotics. Our pets then run around in the park, near a waterway, where they're also now exposed to herbicides at application rates, and they lick themselves. So their gut bacteria are simultaneously exposed to herbicides and antibiotics. Those gut bacteria then find their way into the Avon River. That's my foot. That's not my poo. Mine would be bigger. So a few years ago, we started to look at whether or not there was an effect of other kinds of chemicals on how bacteria respond to antibiotics. And this was reported in The Guardian, the outcome of our first study, which showed that these big herbicides in their commercial formulations cause bacteria to change in their response to clinical antibiotics. And I don't mean any bacteria, I mean E. coli, Salmonella, Staphylococcus, those kinds of bacteria that cause disease in us and livestock and our domestic animals. They did so either by increasing the concentration of the antibiotic necessary to kill them, or decreasing the concentration of the antibiotic necessary to kill them. These herbicides are made up of things other than the active ingredient glyphosate, dicamba, or 2,4-D. You guys know that they're full of things like surfactants. Surfactants are the same kinds of chemicals that in processed food we call emulsifiers. So herbicides not only distribute active ingredient, they distribute food emulsifiers. And we found that those food emulsifiers also cause bacteria to change their response to clinical antibiotics. We measure active ingredient residues on plants. We don't measure emulsifier residues in our food system. They're regulated in processed foods. So for example, the emulsifier polysorbate 80, tween 80, or carboxymethylcellulose, they're limited to something like 0.5 to 1% as a concentration in your processed food, there's no limitation to them in your herbicide. And well below 0.5%, they're causing changes in the minimum inhibitory concentration of these antibiotics of up to 300%. They're not as powerful as the active ingredients, but they're probably more common exposures. They're so common that tween 80, that polysorbate 80, is included in the formulation of the antibiotic in its pill form, ciprofloxacin. So when you take ciprofloxacin, you simultaneously ingest tween 80, which increases the resistance to ciprofloxacin by 250%. Now, we looked at the effect of these kinds of exposures on the evolution of our pathogens. In this slide, what you see is something we call the selection space. So when the herbicide and the antibiotic are together at once, in this case, it increases the concentration of the antibiotic necessary to kill that bacterium. And when we allowed bacteria now to evolve in an environment that had both these ingredients at sublethal concentrations, so this is concentrations at which there's no effect on the phenotype of the bacteria from either agent alone. What we found was that they evolved high-level resistance to ciprofloxacin 100,000 times faster. So the rate of antibiotic resistance evolution was accelerated by the herbicide by a factor of 100,000. Some people say, but wait a minute, you also found that sometimes herbicides make the bacteria more susceptible. So maybe we should drink herbicides when we're on ciprofloxacin. Well, that's an interesting theory, but the problem isn't that they make them more susceptible in a clinical situation. The problem is that they make them more susceptible in an environmental situation. Now, the environment has lower concentrations of antibiotic than do people who have just seen their doctor. And that's on purpose. But if herbicides make you more susceptible to the antibiotic, it means then that those concentrations we more commonly find in the environment are more important for creating the selective pressures that result in the evolution of antibiotic resistance. And that's what's shown here. So you see the brown arrow. The brown arrow is pointing to, really, the figure, the graph on the far right. The graph in the middle shows you the selection coefficient between two strains of bacteria when there's no antibiotic around. And when you go to the right, it's showing you the selection coefficient when you add together an antibiotic and a herbicide that makes them more resistant. The selection coefficient is always favoring the most resistant genotype in the population, always. And it doesn't matter how resistant it is, it just has to be more resistant than the other guy, and it replaces the other guy. We call that evolution. On the left is when the herbicide makes you more susceptible to the antibiotic. When it makes you more susceptible to the antibiotic, once again, the genotype that's resistant to the antibiotic, a little bit more resistant to the antibiotic, always displaces from the population the genotype that's more susceptible. And it does so at many lower orders of magnitude concentration of the antibiotic. So the antibiotic becomes more effective at selecting for antibiotic resistance at lower concentrations. More recently, we've turned to the environment to help us corroborate some of these studies. And we began looking at a bacterium called Serratia marcescens. It's an interesting one to us because it's a pathogen in honeybees. In fact, a recent paper has shown that in glyphosate-dominated agroecosystems, Serratia becomes more likely to kill the bees because it loses biocontrol from the other flora normally in a bee gut. But it's also an opportunistic pathogen of people. So it works for us in both ways, bees and people. We've tested the effects of herbicides and antibiotics on Serratia. And this is the work of a master's student who's just submitted his thesis, so don't talk about it to anybody. Okay? And I just gave you a couple of examples, the effects of ciprofloxacin and streptomycin. And what you see is, in the red, is how quickly the bacteria die when they're exposed to that antibiotic. And you see the red bars tail off fairly rapidly. The blue bars tell you how quickly they die when we add a little bit of the herbicide called number 8, which is the New Zealand glyphosate formulation. Now, you see that it takes much more antibiotic to kill those bacteria. So Serratia, a pathogen, becomes less and less susceptible to the kinds of treatments we might use in hives, particularly in honeybees, to try to control these kinds of infections. A question that we're starting to turn our minds to now is the rest of the world isn't producing antimicrobials to use in people. So plants produce antimicrobials, fungi produce antimicrobials, even other bacteria produce antimicrobials. And all of these antimicrobials probably have an ecological function. It may even be that pollinators and some types of plant pests will choose plants based on antimicrobial effects that they can get, looking for their own therapeutic benefits. So what if herbicides also neutralize the effects of ecosystem-delivered antimicrobials for pollinators and beneficial insects? So that's a question we are hoping to explore in the near future, if by chance one of you leaves your wallet behind. Okay. What this story tells us is not that herbicides are the bad guy, or that food emulsifiers are the bad guy. What it tells us is that they're both the bad guy, and there's probably many more bad guys out there. We just never have tested. We don't ask the question, when does a manufactured chemical to which we may have selective or very common exposures undermine the effective use of antimicrobial agents in an acute fashion, but then also in a long-term evolutionary fashion, because we're losing control of these drugs, and we're losing the control of them at rates that we simply can't explain by out-and-out selection using antibiotics alone. And this graph here shows you global chemical production. The vast majority of commerce in chemicals is from chemicals that have become commercialized since World War II. We have no evolutionary experience with the vast majority of our common chemical exposures, much less how they may interact with other things such as antibiotics, which is also a post-World War II invention. Okay. So the thoughts that I'd like to leave you with are that the environment itself is a place where these antibiotic resistances reside. Antibiotic resistance is natural in many bacteria, but these are not the bacteria that cause disease in us or in any particular target organism that we want to keep alive. Combating this problem will require more than just limiting our use of antibiotics. That simple solution is probably lost to us now. If it were ever true, it's probably no longer true. And the factors leading to this resistance include our other chemical exposures. And if you're all hot and bothered by this and decide you want to run out and cool off in the local stream, remember that our safety assessments do not include any kind of assessment of antibiotic resistance in our waterways. Thank you very much.
No, that's brilliant. Very eye-opening. One question I have is this new synthetic meat that they will get in a lab, make a broth, and they need GMO substances and antibiotics. Do you see that being a long-term sustainable solution for us?
If the alternative is the zombie apocalypse, then yes. Because I would just change my timeline on long term. I think you've identified the key risk issue with these manufactured foods. They are not just pure items, right? They're not the steak that you cut out of, what is it, the ass of a cow. I'm not a farmer, okay? They are a mixture of things. You need, they're an intensive form of protein production as well. And so you need support chemicals like antibiotics. Those antibiotics may not find their way back into the food chain through that manufacturing process, but there will be other ones that do. So my answer is that I don't know that they are going to be a long-term solution, but then again, I don't have a great prognosis for the long term.
I want to ask you about silver stream.
Yeah.
Right, I live in that area. Now, where do you think the problem has come to get into silver stream?
Okay, so silver stream is, I have to also say that the part that we sampled is part of that ecology restorative project area. So it is supposed to be the cleanest part of silver stream that we were sampling in. What do I think is the problem? I don't know. It drains lots of agricultural areas, so it could be runoff into those streams. It could easily be someone leaky home septic tank far enough upstream. All of these are possible. Certainly for the Avon, we have made it part of our forensic reason to exist to track down the source of all the E. coli in the Avon. We will not find all of it, but we did find the predominant source of ciprofloxacin-resistant E. coli is probably a leaky pipe in Upper Rickerton. And we've got it to within about 10 meters, right? That doesn't mean, though, that that pipe being fixed, the problem will go away, but we would expect the levels of resistance to go down. So I don't know, but we won't know until we start tracking down resistant sources rather than just E. coli sources.
In relation to the farming context of farmers applying manure back onto pasture again, as I understand it, there's a stand-down period of 21 days. Do you know how that would, at the end of 21 days, what happens to Salmonella, E. coli, and fecal coliform? Is that enough time to break that down? Because it seems to me that, I don't know, don't get me wrong, I'm not a farmer, but it might sit on the surface for quite a period of time. It sounds like E. coli is going to last longer in the water stream than most people would imagine.
Although we're surprised at how long and stable the populations of E. coli were in water, because we're always told that E. coli isn't naturally adapted to the outside environment, but it does seem to be a self-sustaining population, at least in the Avon River, as well as a new influx population. I don't know the answers to that question, and there will be different practices in different countries. So the statistics that I gave you were from UN reports. They weren't New Zealand specific. Certainly composting your manure before you use it should dramatically reduce the amount of these organisms coming in. But the effect on the DNA of those organisms is less certain. And that DNA can be picked up by other microorganisms in the environment, including other sources of E. coli that might pick that up. So it's part of the solution, but there's probably more study needed to know if it's going to have other pathways. But the other thing I would say is, it's not just through the water that we're going to be exposed. One of the frequent visitors of manure is blowflies and houseflies. And we catch these houseflies and we have them walk across our petri dishes in the lab. And they have little trails of antibiotic E. coli that grow up in their walking trails. They also fly into your homes and land on your dinner. Your pets and pests roam these fields and also have access to your homes. So there's lots of different pathways other than just spraying it on your food.
About 10 or 12 years ago, there was a large study done, I think it was about a $5 million project. It was an insistence by the Green Party that they looked at antibiotic resistance. And I read the entire report. It was quite technical, not many other people did. And I rang the lead author of this, and I said, what's going to happen with this? Are you going to do a follow-up study? And she said, no, there is no money left. And so they've now got a baseline for it. And I said, well, what do you think's going to happen? Do you think things will stay the same, get worse or get better? And being a scientist, she wasn't prepared to answer that. And I said, well, can I reframe it? If I was a betting man, which way do you think I should bet? And she said, I can probably answer that. She said, I don't think it's going to get better. Are you aware of that report, and are they going to do another one?
Well, the Royal Society did do a report on antibiotic resistance in the last couple of years. I was part of that. So that was like 2018 or something like that. And it holds out the prediction that things are getting worse. 2 points make a line.
2 points. Just wondering if you hold out any hope for something like CRISPR-Cas9 as an intervention in this arena.
So could we use those as antimicrobial agents? Is that your question?
It even somehow negate the problem in that particular strain of bacteria.
I think that if you already had control of the bacteria to the point where you could introduce CRISPR-Cas9, then yes, but that will be after the patient's dead. But I'm not making light of your question, right? But there are formulations coming out where people are using things like these nucleases to spray directly on plants, people, other kinds of animals.